Welcome everyone, thank you for joining the company presentation of Bridge Biotherapeutics. My name is Pavel Princev and I'm the Director of Business Development. So starting off with a quick company overview, our company is operating out of two geographies, in South Korea and in the United States. We were originally founded in 2015 in South Korea, and the difference between the two centers is our South Korean research headquarters are more focused on the R&D for our core pipeline, and they set the strategy and governance for the organization as a whole. The Boston Discovery Center was established not too long ago, around the end of October of 2020, and they have their own in-house development program focusing on early-stage oncology assets. So a little bit about who we are, where we are now, and where we see ourselves in the coming future. Originally, when we were founded, we had a very, very small team that in-licensed 3 first-in-class assets that the team brought through IND approval in the US, and then eventually we had our IPO in South Korea in 2019. Right now, we're about a year or so in our growth period, and we have already accomplished our goal of establishing the Boston Discovery Center that I mentioned earlier, and our remaining goals for this period is to increase our clinical pipeline to 7 clinical programs. We are approaching 4 clinical programs at the moment, and to have our first FDA marketing approval, and then also increase our employee headcount to about 100 people. We are at close to 50 people at the moment. From 2026 onwards, we strive to become a global commercial stage company with an established sales network in the US and having close to 500 employees by that time. What I'm going to be talking about today is our clinical pipeline and late stage pipeline in the order that you see here from top to bottom. Starting off with BBT-401. BBT-401 is a first-in-class polino-1 protein-protein interaction inhibitor for the treatment of ulcerative colitis, a debilitating disease in which there is inflammation in the colon, severely affecting the patient's quality of life. Toll-like receptor signaling is well known in the pathogenesis of several inflammatory diseases, with UC being one of them. And BBT-401's novel mechanism of action works through the disruption of polino-1 containing MyDA8 and RIP1 complexes. By disrupting the formation of these complexes, downstream NF-kappa beta is unable to translocate into the nucleus and cannot initiate the induction of pro-inflammatory genes that are most often associated with the disease. In the middle preclinical efficacy chart here, DSS-induced colitis mice were given BBT-401 at 3 dosage levels, low, mid, and high, and we were able to see a clear dose dependency across the 3 dosage levels. Also, compared to the competitor, here, the mid and high dosage groups were able to show a significant reduction in disease severity in that model as well. And then on to the right side here is our initial clinical efficacy data from our low-dose cohort in part 2A. So over an 8-week period, patients were given our gut-restrictive oral formulation of BBT-401. And efficacy was measured using the industry standard Mayo score. We were able to see reductions in the Mayo score in several of the treatment groups, as well as a decline in one of the key disease biomarkers when compared to control. Right now, the phase 2A low-dose cohort study has already been completed in the US, and we are currently conducting the phase 2A in the medium and high dosage groups globally in the US, Korea, New Zealand, and in Eastern Europe. So recruitment is completed. So far, the patient recruitment is done, and we're expecting a detailed clinical study report towards the end of this year. Additionally, the phase 1 China ethno-bridging study in healthy volunteers was also completed in January of this year, and we are expecting the CSR sometime in the 2nd quarter or 3rd quarter of this year as well. On to BBT-877. BBT-877 is our first-in-class autotaxin inhibitor for treating idiopathic pulmonary fibrosis. Autotaxin is an enzyme that has been found in higher concentrations in patients with various inflammatory and fibrotic diseases, including fibrosis of the lung. Autotaxin is an enzyme that catalyzes the hydrolysis of LPC as well as the local production of LPA. In turn, that LPA then binds with and activates several LPA receptors, triggering a downstream signaling effect leading to epithelial apoptosis, which is one of the primary initiating pathological events leading to the IPF, mottled pulmonary fibrosis, as well as interstitial pulmonary fibrosis. On the right-hand side here are charts displaying the LPA levels in patients that were given BBT-877 in a multiple ascending dose from our phase 1 trial. On the left-hand side, we can see in QD dosing, a greater than 80% reduction in LPA levels has been observed from the 800 milligram dose. And on the right-hand side, we were able to see 80 and 90% LPA reduction levels from the 100 and 200 milligram dosage levels, respectively. In terms of development for this asset, phase 1 is done. We are now in discussions with the FDA regarding our clinical trial design and plan for starting the phase 2. We hope to start that globally by September of this year, starting in the US, followed by entering countries in Europe and in Asia. Also, the DDI study in healthy volunteers is planned to be conducted toward the end of this year. And then on to our oncology assets, BBT-176. It is our first-in-class and best-in-class fourth-generation EGFR tyrosine kinase inhibitor for non-small cell lung cancer. When it comes to metastatic non-small cell lung cancer, one of the most dominating genetic drivers of this disease is mutations in EGFR. And what we see, the deletion in exon 19, del19, and a point missense mutation of L858R comprise about 90% of all of the EGFR driver mutations. So first generation and second generation EGFR TKIs were developed to address these driver mutations. However, as many of you probably know, the emergence of the T790M mutation renders the majority of these drugs ineffective. So shortly after, we started to see the emergence of the third generation EGFR TKIs. Osimertinib was one that's well known, had very promising clinical efficacy results leading to its approval in second line following first and second generation EGFR TKIs. And shortly after that, it was also approved in the frontline setting as well. The similar story with what happened with the first and second generation EGFR TKIs. Within about 10 to 12 months, the majority of patients that were taking osimertinib eventually progressed on that drug as well. One of the most prevalent known on-target mutations that is responsible for the progression of osimertinib is the emergence of a C797S mutation. There is currently no viable treatment options for patients that are presenting with this mutation, and BBT-176 was the world's first fourth-generation EGFR TKI to enter the clinic in South Korea in 3 sites, and that was in April of last year. So on to some of our interim clinical efficacy results. Here is showing our ongoing dose escalation data from 20 milligram to 480 milligram cohort. Within this subset, we have been able to see 2 cases of partial respondents. The first patient who had a partial response was someone who has previously been on gefitinib. They presented with a del19 driver mutation, and we were able to see a 30% reduction in that particular patient. Our second PR case was a patient who progressed on osimertinib and presented with a C797S containing triple mutation, and that patient had a 51% reduction in their tumor size. Furthermore, the patients that have been taking this drug are responding very well to the drug. There has been no serious adverse events. There have been no DLTs up to the 480 milligram cohort. The majority of the patients, as you can see here, have been on the drug for over 200 days, and some of them are approaching a year on the drug. So where we are now, we're almost complete with our phase 1 dose escalation study. If any of you are going to be attending the WCLC in Austria in a couple months, we will be there presenting our updated phase 1 data on this asset, so please come by and say hi. Right now we're going to be having our end of phase 1 meeting relatively soon, where we will be figuring out the final details of our clinical development plan, as well as determining what a recommended phase 2 dose will be, and we plan to initiate our part 2 dose expansion study in the United States early next year. And then finally, our other fourth generation EGFR TKI, which is BBT207, is a structural derivative of BBT176. BBT207 has been developed with an enhanced activity profile for C797S containing mutations in the double mutant setting, as well as being demonstrated potential in a frontline setting. We believe that BBT207 is ideal for patients who have progressed on osimertinib from the first-line setting. And then some of our preclinical data here for this compound, we saw that BBT207 has been able to induce strong tumor regression in all of the tested mutant models that we had, from dosages between 20 and 40 milligrams per kilogram given orally once daily. Our timeline for BBT207, we expect to have the IND submission done toward the end of this year, and we will be entering our global phase 1 study for this compound also towards the early next year, ideally in the United States, starting from the United States. And this concludes my presentation, so thank you very much everyone for listening to my talk. And if you have any questions on our programs, as well as would like to discuss collaboration and partnering goals, I'm here and I can meet you outside. Thank you very much.